Good morning. Welcome to my class, and this is BIO101 Chapter 15 Chromosomal Basis of Inheritance. As usual, the first two slides I would have listed out all the important topics that we would be discussing in that particular chapter. Okay, this was the time of 1860, and Mendel's observations were put forward. People were aware of the Mendelian ratio of monohybrid cross, dihybrid cross, the Punnett square, and the different inheritance patterns that he drew a parallel from the pea plant, and yet people were still skeptical. So I would just like to go over the meiosis and put it in perspective of Mendel's laws and talk about how homologous chromosomes behave and the process of how segregation takes place and how non-homologous chromosomes behave, and it talks about the way they assort independently and so on. Okay. So for that, a little bit about meiosis, and what I wanted to talk about in this is, so if you are a person with say 6 chromosomes, a theoretical person, so every cell has 6 chromosomes. This actually means 3 chromosomes come from your mom and 3 chromosomes come from your dad. So let me call the 3 chromosomes as M1, M2, M3, and the 3 chromosomes from the dad as D1, D2, and D3. By this, what I am trying to say is M1 and D1 are homologous. Which means these chromosomes will have the same length, same staining pattern, same place for centromere's position, and will have genes for the same qualities. So homologous chromosomes, meaning if this particular gene had this particular chromosome have gene for say hair color, eye color, and say tallness, the same M1 will have and the same D1 will have for hair color, eye color, and tallness. So it goes to say it is not identical, but it is similar. Not identical. And during meiosis, which takes place only in the gametes or the cells destined to become the gametes, like the ova or the sperm. Okay, during meiosis, it is divided into a series of stages called prophase 1, metaphase 1, anaphase 1, telophase 1, and then a cytokinesis, and then prophase 2, metaphase 2, anaphase 2, and telophase 2. So in this, the unique patterns are in prophase 1, the homologous chromosomes, which are duplicated, align themselves along the entire length, which means let me just get this out of the way so I'll have some space to write and draw and explain what I was trying to say. Just give me a minute. Okay, and Okay, so what he basically says is the M1 has got two copies. So I have M1, M1, and then the D1, D1. So the M1 and then the M1, these two are sister chromatids. And then if I need another color, this is my D1, this is my D1. So these guys align themselves along their entire length, end to end, looking like a tetrad, and there is crossing over and exchange of DNA. Now by that I mean, supposing the M has a quality for tallness, and so quality for tallness, and dad has the quality for shortness, or say kind of like this. So, and I'm just taking, this is a hypothetical situation, and here maybe this is eye color brown, this is blue eye color. Okay, so there is a likelihood of a crossing over which can lead to, this is T and this is brown, this can be T, that is the small t, and still brown, and this can be blue and this can be a capital T. By that what I mean is, this part of it there has been an exchange, and this part of it there is. So you see these two genes have kind of exchange chromosome. Now which one I mean, this one and this one has kind of exchanged themselves in this particular stage. Okay, so that is where you will have an exchange. And then you have metaphase 1. During metaphase 1, the way it works is, okay, the alignment. By that what I mean is, the chromosomes align themselves along the equatorial plane of the cell. All right, so in this particular case, if the chromosomes, then means this M1 and the other copy of the M1 is on this side, D1 and D1 is on this side along the plane. So these guys are like a tetrad, like you can see here. Okay, and then I can still have M2, the two copies, and the D2, exactly the way it is here, and then I can have D3, and M3. By this, what I am trying to say, there is no hard and fast rule to say all of the mother's chromosomes should be on one side and all of the father's chromosomes should be on the other side. The alignment is independent. The alignment is independent of the other. It does not, one chromosome set does not have a say on how the alignment of the other chromosome set could or has to be. Okay, so that is part of the speciality of the meiosis, which we know now, but we already kind of figured it out, or Mendel kind of figured it out through his experiments. So the law of independent assortment kind of falls in place here. Okay, then another interesting thing is the law of segregation. Now by segregation what I mean is, it then finally decides that which chromosome goes into which cell is really not a specific pattern, whereas it is a chance or a probability how it can occur, which you will see now in the next slides as we go through the whole process and talk about the laws along with it. So here what he is talking about is, this is the F1 generation, and he is talking about two genes or two characteristics that he is talking about, which is round seeds and then wrinkled seeds. Round is a capital R, wrinkled is a small r. And then yellow colored seeds, which is capital Y, and green colored seeds, which is a small y. So the interesting part was they were present in different chromosomes. So you can see that they are present in different chromosomes. In this case, the two blues that contain it, you can say that it is being contained in the chromosomes of 1 and 2, which means the M1 and M2, for example. So M1 contains the quality, say, of Y, and the M2 contains the quality of, say, R, and D1 contains the quality of small y, and D2 contains the quality of small r. So this particular cell is a combination which contains what the mother has given and the father has given, and these are the two copies or the alleles, and these are the two different genes present in the different chromosomes. All right. So during metaphase, the alignment can be any which way, meaning all mother's chromosomes on one side, all father's chromosomes on the other side, or it can be a shuffle. One of mothers, one of fathers, one of mothers, one of fathers. So this is the part he's talking about when he explains the law of independent assortment. Okay, so then you would have a further step wherein in anaphase, they kind of anaphase 1, that is, okay, they kind of distribute themselves in such a way that one cell, now in this case it is the M1 and M2, but two copies of it, and in this case it is the D1 two copies and D2 two copies, or in this case he says it can be M1 two copy and D2 two copy, and then this can be D1 two copies and M2 two copies. So he's trying to tell you that the assortment has taken place in any which way. You can either have all of dad's chromosomes or all of mom's chromosomes, or you can have a mix of them and it has kind of gotten shuffled. Okay, so in the metaphase 1, you can have the segregation or the shuffling, and it can have any which way the combination that you want it. In anaphase 1, the alleles segregate, meaning when there was a situation wherein the alleles were two, let me just go back, excuse me. Okay, so in this particular case, if you notice, the alleles segregate. So what I mean by that is, if I have in my M1, okay, I have, say, capital R, and in my D1 I have small r. Okay, at this particular part you will see that when they kind of stretched and went apart, you just had one kind at the end of it. You had either a capital R or a small r, like they segregated out. In this there was a combination, they segregated out. So the homologous chromosomes M1 and D1, it's finally decided that only one kind can go into a cell. All right, so the law of segregation fits into this, and law of independent assortment also fits into this. So my point at the end of this explanation is the two laws that Mendel had come across and put forth falls in place when you look at the entire process of meiosis. Okay. Now we move on to talk about another scientist. His name is Thomas Hunt Morgan. He was an embryologist. He also started working with fruit flies. Now, why did he use fruit flies for his experiment? There were certain good qualities that were useful in genetic studies. One, they were very common. They would feed on fungi growing on the fruit, so then it was easy to find food for them. They bred at a very high rate. A generation can be seen every 2 weeks, and they only had 4 pairs of chromosomes, which means it might have M1, M2, M3, M4, D1, D2, D3, D4. This is how it might be having its chromosomes, whatever that it got from its parents. So, in the process, for 2 years, he kept breeding them, and all of the fruit flies ended up having only red eye color. Red eye color was the only thing that they saw. Suddenly, one day, when he was almost ready to give up, he noticed one fly with white eye color, and he was feeling extremely rewarded, extremely ecstatic about the whole process. So, the genetic makeup that you would normally see, meaning the normal phenotype that you would see always, or what that which was common, is called wild type, and they labeled it as W plus. Okay, the traits alternative to this wild type, meaning something that differed from the usual, is called a mutant and a mutant phenotype. You can just call it W, or you can call it W minus, in order for you to remember that the minus indicated for you, you can do it, indicated that it is a mutant. Okay, so now this was a totally different scenario. So far, when Mendel was dealing with chromosomes and looking at the different qualities, he was just looking at the pattern which was common to both male and female. This kind of made the story slightly different, and Mendel kind of figured it out that this particular inheritance patterns is not like a same hand-me-down for the male, same hand-me-down for the female. The pattern of inheritance differed in males and in females. You will see in a few slides from now how he comes to prove that the inheritance patterns differs and why. Okay, so the first thing was he started with a red-eyed female. Now female, it is known that they have 2 X chromosomes. Okay, I'm filling here. Male, it is known that they have 1 X and 1 Y chromosome. Okay, so he had a red-eyed female and he crossed it with a white-eyed female. Now red-eyed female, what I am trying to say, in the parental generation, for always, it has always been red-eyed females giving birth to red-eyed females, and so on. Purebred in the parental generation. White-eyed male, that's it. Okay, and then in the F1 generation, all of the flies had red eyes. And in the F2 generation, he noticed that 50% of the males were red-eyed, 50% of the males were white-eyed, and all females were red-eyed. And how does this work? Let's see. So, in the parental generation, he started with a red-eyed female and a white-eyed male. In the F1 generation, everyone had red eyes. So the inheritance pattern kind of worked in such a way. After that, he kind of crossed this red-eyed lady and a red-eyed male from this generation. Okay, one second. Let me just go back and okay. So here, if you notice, in the F1 generation, all of the females were red-eyed and male was white-eyed. Okay. Now, the conclusion was in the parental generation, the lady who was offering her chromosomes, both her X chromosomes had the wild type gene in them, whereas the white male who was offering his chromosomes, he has only one X chromosome and he was offering a W minus or a mutant variety. So if we take this white-eyed male and cross it with a red-eyed female, you would see that the way it works. Let's just do it here in a Punnett square so you kind of get the hang of what I'm trying to say. The X plus, this is the female, all right? And then X minus and Y, this is the male, all right? So X plus, X minus, X plus, X minus, X plus Y, X minus Y. So in this, as I had explained verbally, all of the females looked red-eyed, 50% of the males looked red-eyed, and 50% of the males looked white-eyed. Okay. In the F2 generation, on the other hand, when they crossed this with a red-eyed male, all right, the carrier, meaning the second generation, but he was very careful to use these two guys, which means a red-eyed male and a red-eyed female. But the red-eyed female here is a carrier, and if you put them together, what happens here is, let's just put that and see, X plus and X minus, and this was X plus Y. So you see X plus X plus X plus X minus X plus Y, X minus Y. Yeah. So in this case, you see one white-eyed male at the end of the Punnett square in the F2 generation. This kind of helped make a connection that the genes that are present in the X chromosome have a different mode of transfer to the next generation. The reason being, females have two X chromosomes, and the way they would exhibit an expression of a certain gene would depend on what is present in each of those X chromosomes, right? Whereas males have only one X and one Y. So whatever quality they exhibit is because of what is present in that one lonely X. They do not have an alternative, all right? So the chromosomal basis of sex, basically X chromosome and Y chromosome. Now remember, during meiosis you have crossing over where the homologous chromosomes kind of come together and bond along the length, right? In this case, you have to remember, if it was a female, okay, so this is one X chromosome, okay, so I'm going to call this one X chromosome, and this is the other X chromosome, all right? They can kind of sort of align themselves along the lines, and there can be an exchange that can take place, all right? But on the other hand, if it is an X and a Y, so think about it like this, if there is an X and a Y, okay, which means this X chromosome is larger, the Y chromosome is kind of very small, so there are only certain parts of it which are homologous, and yet there can be some amount of crossing over still taking place. So that process still continues of the prophase 1 in meiosis. Now, what makes the Y chromosome special or unique? It has a gene, it is called SRY gene, and it translates as sex determining region of Y. So this particular chromosome is helpful in determining the maleness or the qualities that go to determine that particular human being as having male qualities. Okay, so this is your X chromosome. And these, remember, these are two sister chromatids, okay? So this is one X chromosome, this is another X chromosome. This is one Y chromosome, and this is another Y chromosome, the sister chromatids of it. So this part of it and this part of it perhaps are like homologous, and they can still align themselves as a tetrad for exchange in that case also. Now, there are different ways in which you would have inheritance, the sex determining patterns. We, for this chapter, are following only the XY system as followed in humans. Alright? So, here you see the sex chromosomes have genes for many characters unrelated to the sex. So a gene located in either sex chromosome is called a sex-linked gene, or it is also called as an X-linked gene. Now, the other characters that are unrelated to sex that are present is one particular disorder called hemophilia. It is a sex-linked trait. And you would have another quality that we talk about is color blindness. So they follow a specific pattern of inheritance. Remember, fathers will pass all their X chromosomes to their daughters and none to their sons, because they are passing the Y to the son. Alright? The mothers can pass on to both daughters and sons the X chromosome only. So remember, another important note is Y chromosome is passed intact from father to sons, and that is why even before people knew specifically the details about X chromosome and Y chromosome, they kind of knew that the son and the grandson and the great grandson and the great great grandson kind of can take the Y chromosome intact, and that has probably been the basis to having a male heir to the kingdom or to the property and so on and so forth, to carry the family name. Okay. For a recessive sex-linked trait to be expressed, recessive means it needs 2 copies. Alright? The female needs 2 copies of the allele, whereas the male needs only 1 copy, because that is the only copy that exists. Because he is an X and a Y, if he has one defective gene in his, just a second, excuse me, in his X chromosome, he's bound to express it. He doesn't have an alternative with which he can kind of make do. So sex-linked recessive disorders are much more common in males than in females. Okay, so let's understand with a few examples how this is done. Okay, so here in this case, this is a female and she is normal. Both her X chromosomes are with a capital N, which says she's normal. Alright. In this case, the male is affected. So I'm kind of putting his number or name kind of like this. So if you notice, this is an X minus and Y. This is an X plus. This is an X plus. So I'll have 2 daughters. If you see, they are not in a dark orange color, neither are they in a white color. They carry the gene, but they are not exhibiting the traits of that gene, because they have the other good X gene, which kind of makes them seem normal. Alright, so there is 2 carrier daughters. Okay, one normal son and the other also normal son. So you notice the father had the defective gene. He did not pass it on to his sons but to his daughters. Alright. On the other hand, if we have the next instance or example to look at, and work it out. We have a carrier daughter, X plus and an X minus, and a normal son. Alright, so this is a plus, this is a plus and a minus. This is a normal healthy girl, normal healthy boy, a carrier girl, and an affected boy. And this gene is passed on by the mother to the son. Alright, so there are different ways in which you can say this or express this. In this case, for example, 50% of their children were carriers. You can say 50% of their children were healthy. Or you can even say 100% of their sons were healthy. 100% of their daughters were carriers. Do you see how you can express it in different ways, but it means this? Yeah. On the other hand, let's see how this one works out. In this case, you can say 50% of their children are healthy. In that, 50% of their daughters are completely healthy, normal. 50% of their daughters are carriers. 1/4 of their children are carriers. Or you can say 1/4 of their children are affected. Or you can say 50% of their sons are affected. Yeah. So there is different ways in which you can express the same set of results. So it's a good idea to know that in order for you to be able to understand what exactly is being asked, and so that helps you answer the questions in the exams. So a good idea is to kind of write it down and draw what kind of genotype that person has. Okay. Now, in the third example that we have, in this case, he says the daughter is a carrier and not the daughter, I apologize. The mother is the carrier and the father is affected. So in this case, if you notice, I will have an X minus and then a Y, and then I will have an X plus and an X minus. In this case, I have one carrier daughter, one affected daughter, one affected son, and one normal son. Okay, so these can be expressed in different ways. 50% of their children are affected. 50% of their sons affected. 50% of their sons normal. 50% of their daughters affected. 50% of their daughters carriers. 25% of their children are normal. Okay, so this explains how you can have the chromosomal inheritance patterns of X-linked disorders or X-linked genes. In addition to this, we have something known as X inactivation in female mammals. Now, what does that mean? It basically means females have 2 X chromosomes. So each of the cells will end up having 2 X chromosomes. We don't need 2 X chromosomes. So at certain stage of embryonic life, certain cells kind of inactivate one of these X chromosomes, and it stays quietly as a ball, as a tight condensed ball in one corner of the cell, and the other one stays active, or the other one, is inactivated and this one is active. So this inactivated condensed X chromosome that is present in a corner of inside the nucleus is called a Barr body. Okay, this occurs randomly and independently. Now, females are generally a mosaic with two types of cells. The example that we can use to explain that is sweat glands or tortoise shell cat. I would like for you to watch a video in order for us to understand that.
Inside each one of your cells, there is 6 feet of DNA, made up of 6 billion letters of genetic code. Now, your DNA is split into 46 pieces, each 3 to 4 centimeters long, called chromosomes. Now, normally we think of chromosomes as looking like this, but they only take that form when a cell is ready to divide. So usually, DNA is just a wiggly thread within the nucleus. Now, if you can imagine, DNA is only about 2 nanometers wide, but a chromosome is centimeters long, so you would think that it would get tangled worse than the headphones in your bag. So the DNA is actually wrapped around proteins called histones. Now, those histones have wiggly tails, which will come in handy, as we'll see in a moment. Your unique set of DNA first formed when 23 chromosomes from your mom mixed with 23 from your dad. Now, 22 of those chromosomes from each parent form matching pairs. But the 23rd set is the sex chromosomes. So 2 X chromosomes make you female, and an X and a Y make you male. Now, since the male sex chromosomes are different, both can remain active for the rest of your life. But for females, one of the X chromosomes needs to be inactivated in order for proper development to occur. This happens when a female embryo is just 4 days old and consists of only 100 cells. Right now, in this cell, the X chromosome from dad and the one from mom are both active. But through a tiny molecular battle, one of the X chromosomes wins and remains active, while the other is inactivated. This is done by packing the DNA closer together and making modifications to those dangly histone tails that signal this inactivation. New structural proteins are also added to bind everything closer together. And finally, methyl groups, these tiny little molecular markers, are added to the DNA to basically signal to the cell that this DNA shouldn't be read. So all of this together makes the DNA very difficult to access for the molecular machinery that would harness the code in this DNA. It is switched off. This DNA is silenced. In contrast, the active X chromosome DNA is more spread out. This allows better access to the genes on the chromosome. Histones can be slid along the DNA or removed entirely. And the histone tails have a different modification, signaling this DNA is active. Now, all of this makes it possible for RNA polymerase to access and transcribe this DNA into messenger RNA, which then goes out into the cell and is used to make a protein. Now, what's surprising about X chromosome inactivation is that it seems kind of random which X chromosome wins. I mean, in some cells, dad's X chromosome wins, and in others, mom's X chromosome wins. So this 100-cell embryo ends up with a mixture of active X chromosomes. But from this point forward, as these cells divide, they maintain the active X chromosome that they had inside. So all of the cells with dad's active X chromosome give rise to further cells with dad's active X chromosome. And this continues on into adulthood. So if you could look at a woman's skin and see which X chromosome has been inactivated, you would see a stripy pattern, which shows the growth and migration of all of these first 100 cells when the embryo was just 4 days old. Now, of course, you can't actually see that in humans, but you can see this with calico cats. And that's because the gene for coat color is actually on the X chromosome. So just by looking at the pattern of her spots here, her dark and light spots, you can see where her mom or dad's X chromosome has been inactivated. And this also shows us that only female cats can be calico cats. And that's because, well, only female cats can inherit 2 X chromosomes with 2 different color genes. Now, this is just one really cool example of epigenetics. But epigenetics doesn't normally work on one whole chromosome. In fact, it's at play in all of your chromosomes, turning on and off your genes. For example, it's epigenetics which makes a pancreatic cell capable of producing insulin, because that gene is switched on there, but switched off everywhere else. What's more interesting is that it seems the behaviors you take can actually affect your epigenetics. And even weirder, perhaps the things that your parents or grandparents did can affect your epigenetics now, can affect your DNA. So you are not just a product of your genetic code. You're not just a product of your DNA. You are also a product of your epigenetics. And that is influenced by your behavior and the behavior of your ancestors. Inside each one of your cells, there is 6 feet of DNA, made up of 6 billion letters of genetic code. Now, your DNA is split into 46 pieces, each 3 to 4.
Okay. Sorry about that. I hope that video was useful in understanding what is bar body and what is mosaicism. And the examples that they had used for us was the tortoise shell cat or the calico cat, and in human beings the example is the sweat glands, these genes being present in the X chromosome. Okay, so a visual representation of how this inheritance pattern works is there are 2 X chromosomes. One is an allele for an orange fur and the other one is an allele for a black fur. And during cell division of this particular embryo, when it is about 100 cell stage, one X chromosome in some of the cells get inactivated and the other X chromosome gets inactivated, and this one gets expressed in certain, oh, just a minute, let me just go back, excuse me. This one gets expressed in certain cells, and this color gets expressed in certain cells, giving you a pattern of black and orange colors in tortoise shell cat, or a black and white color pattern in a calico cat. Okay, now each chromosome has hundreds and thousands of genes, and the gene present on the sex chromosome is called the sex-linked gene. Okay, now there is another concept that we are going to be discussing. That means linked genes. Linked genes means 2 or more genes that are present in the same chromosome that tend to be inherited together. Now, normally in my class, I would always have like a pair of students who always function together, meaning like they are very good friends or best friends. So wherever one goes, the other one will also follow. So I kind of use them as an example to say these are like linked genes. So then there used to be one boy named X who would be present in my class, and he was friends with another boy named Y. So X and Y, or say A and B, you can call them anything, they would be linked together, meaning they are attached or inherited together, and they would go along together. They do not assort independently. They are very, very dependent on their movement. Okay, and so during a dihybrid cross, you will not get this kind of ratio. You will kind of get the monohybrid ratio. What I mean by that? One second. So think about it. I normally use ice cream sticks in my class to explain this particular phenomenon, but just so we understand each other. So supposing I have an ice cream stick which says capital A and capital G, okay, and I have another one which says capital small a and small g. Okay, so this is what this particular person has. Now when he does segregation in meiosis, one of his cell will have the chromosome which has capital A and capital G, and the other one will have the chromosome saying small a and small g. You notice, because this is because both of them are present in the same chromosome. They are even present together, perhaps for them to be assorted together. So they are like they do not follow the pattern of independent assortment, they follow a pattern of dependent assortment, which means they will have the same kind of ratio as 3:1. Okay, instead of the 9:3:3:1 ratio that you would see if at all you would have them as in different chromosomes. For example, let's just explain that bit also. So supposing this is chromosome M1 and which has capital A. This is chromosome D1 which has small a. This is chromosome M2 which has capital G. Okay, and this is chromosome D2 which is got small g. Now when these guys assort in meiosis, I can have 4 different combinations how they can assort. Okay, one could be I can have capital A, capital G, or I can have capital A, small g. I can have small a, capital G. I can have small a, small g. So you see, this shows where this guy goes, this does not have to follow. It can be this guy. Do you see the assortment in this particular case where they are not linked, where they are present in different chromosomes? Those genes tend to assort independently and give you the ratio of 9:3:3:1. If you need to know further about this particular assortment, you need to go back and look at the video on Mendelian genetics one more time. But I think I clarified the point for fairly easily or fairly understandably for my students. Okay. So now in this particular Mendel's test cross where he was trying to do, now we are going to try to do one test cross where you expect equal expression, especially of these guys. So what he is trying to say is, if I have this as my combination, remember this guy is always going to give you small y, small r. So I'll have only small y, small r. Whereas these guys are going to give me so many different combinations. So you see, I can have capital Y, capital R. That's one in one of the haploid ova or sperm. Capital Y, small r. Small y, capital R. Small y, small r. So you see, I can have these combinations. Capital Y, small y, capital Y. Kinda like this. Okay. Capital Y, small y, R. Y, small y, capital R. Small y, R, R. So you observe in this particular case, this one looks exactly like the parent, and this one looks exactly like the parent here, and these two are totally different and unique from the parent. They are like a combination. All right, so now this particular distribution occurred in this fashion because the genes that we are talking about are in different chromosomes. Okay, so that is what he is talking about when he says recombinant and parental type. Parental type means exactly looking like the parent, and recombinant type means he's like a combination. Whatever I did in the previous slide, it is done along with a picture, so you kind of are able to put all of that in a pictorial form and visual form and understand it. Now we follow the same understanding in Morgan's experiment, where he worked with Drosophila flies and kind of worked out to find out the main end point to this entire experiment was crossing over pattern was discovered. Till then, people probably did not know that crossing over happened. They only knew that you can have these different combinations, and with these combinations you can have different varieties, and further varieties can be produced because of crossing over. And crossing over occurs very commonly during prophase 1 of meiosis, and some genes which are close to each other tend to be crossed over along together, or because they are linked they would go in together. And other genes which are very far away from each other, the recombination frequency is kind of 50-50, which means it's almost as though they are in different chromosomes. Okay, so let's have a look at what the experiment was and how the conclusion came to be. Okay, so in this case he is talking about two different characteristics. One was body color and the other one was wing shape. So body color, there was gray and there was black. Wing shape, there was normal and there was vestigial. Okay, so he has two parental types and he introduces gray wings and normal body, gray body and normal wings, I apologize, and black body and stubby wings. And this one that he has introduced is heterozygous for it. Alright, so let's see how he ends up getting the different patterns. This leads to the conclusion, whatever that he was seeing, that the genes were present in the same chromosome and that they were linked. But when he kind of crossed these guys together, he got these two as their large number of offsprings. But he also got certain anomalies, or like certain recombinant which did not fit what the parents looked like, and that frequency in which they occurred was about 17%. And this kind of laid a foundation to say we can do this kind of experiments. We can find out how many recombinants you get, and this way we can find out what is the recombination frequency. This way we can kind of map the genes, or find out in any chromosome where are the genes located, close to each other or far away from each other. Okay, so he kind of have like an ordered list of loci along the chromosome. So higher the recombination frequency, further apart the two genes are present in the chromosome. The genes that are close together are likely to move together. This is the principle that is used to map the genes on the chromosome. So you can have something known as a genetic map or a linkage map. Okay, this does not exactly measure and tell you this much distance away, but it kind of tells you which one comes first, which one comes next, which one comes follows that after that. Map units are kind of like 1% recombination frequency. That is one way of giving you a sense of the placement. Okay, so our experiment starts with a gray, I would like to call it capital G, and normal, capital N, okay, and here I would like to call it small g, and I would like to call it N. Okay, this is what he started with for his parental generation. Alright, so when he crossed them, he got offsprings which were capital G, small g, capital N, small n, because all of the sperms or eggs provided by this creature will have only capital G and capital N. All of the sperms and eggs provided by this creature will have only small g and small n. So the offspring that is out of the outcome of these two parental union will be capital G, small g, capital N, small n. But outwardly they would look grey and normal. Okay. Then he took this and he crossed it with the parental type. So he kind of took the capital G, small g, and double muted once again. So he took a capital G, small g, capital N, small n, crossed it with again small g, small g, small n, small n. At all times, his meiotic division is going to yield small g, small n. Here I'm going to have 4 combinations. Okay, let's see. So capital G, capital N, capital G, small n, small g, capital N, small g, small g. This is what I'm going to get. Okay, let's see what happens in the next slide. So he kind of crossed them the way I was describing in the previous slide. Okay, and in this test cross, let me just go back. Okay, in this test cross, okay, you would have this guy is small g, small n, right? This is capital G, capital N, capital G, small n, small g, capital N, small g, small n. Okay, so you would have capital G, small g, capital N, small n. This is like the parent. Okay, like this particular parent. Then you could have capital G, small g, but small n, small n. This is representing neither parent. Small g, capital N, small n. This is representing neither parent. Small g, small n, small n. This is representing this particular parent. So he's got 2 parental types and 2 recombinants. Now, the important question to ask in this: are these 2 qualities or characters present in the same chromosome? If they were present in the same chromosome, I would not be having this combination or this combination. I will have only this possibility and this possibility, meaning I will be, this particular creature will be capital G, capital N, small g, small n. Yeah, so I will be able to give during meiosis this kind and this kind in my sperm or in my ova, depending on whether I'm the female or the male. In which case these 2 chromosomes have the 2 qualities together in, or they are in the same chromosome. All right, if they were present in this combination, please bear with me, this is mother, this is M1, this is D1. Okay, supposing it was this, I would have had only this combination and this combination, not these 2 combinations. But supposing it was M2, D1 and D2, supposing this was capital G and capital N and small g and small n, I could have so many combinations. This one, this one, this one, this one. So I will have an equal number of all of these types, parental types and recombinant types. Now let's see what the actual result gave us. I repeat, if I had a situation where these 2 characters that I am talking about for body color and wing size were present in the same chromosome, I would have a distribution or segregation in this pattern where I have only two parental kinds of assortment, and I would have had only these two qualities as my progeny or my babies. Or option 2, if I have them in two separate chromosomes, my distribution pattern and the segregation during meiosis will give me these 4 combinations. So I will have equal number of 4 types of these progeny. Alright, so this is exactly what I had written in letters. Let's just rewrite in letters so we are clear. So this is small g, small n. This is capital G, capital N. This is small g, small n. This is capital G, small n. This is small g and capital N, which gave me capital G, small g and n. Okay, for this one. Then it gave me capital G, small g and small n's for this. And then for this it gave me small g, small g and I'm sorry, let's just go back. I think I flipped this here, but I think people understand what I'm talking about. Let me just do this once again. So in this one I have capital G, small g, n and n. In this case, no, sorry, this should not have been here like this. This should have been a small g. I apologize. This should have been this way. I mean, he is given this kind of distribution. Okay, so small g, n, n. So this is another parental type, and this would have been, okay, capital G, small g. Okay, let me rewrite the whole thing because it does not match with what I wrote in the previous one, though the genetics of it is the same. I apologize. Let me just go back and write this. So small g, small n. So this is capital G, capital N. This is small g, small n. This is capital G, small n. Okay, and this is small g, capital N. That's what he kind of flipped them. Okay, he wrote the two parental types together here and the recombinant types together here, whereas I had written it like one parental here, one parental here, and two recombinants in between. All right, so now in this case I would have capital G, small g, capital N, small n. So this is one parental type. Small g, small g, small n, small n. This is another parental type. So this resembles this parent. This resembles this parent. Okay. Then I have capital G, small g, and n. This represent neither parent. And then I'll have small g, capital N, small n. This represents neither parent. This is my recombinants. Now, like I said in the previous slide, if they were all qualities or characters in different genes, I would have an equal number of all of them. If they were qualities present in the same gene, then I would have only the parental type and not the recombinant type. What did I actually get? Let's see. So, this is what I get. If they were on the different chromosomes, okay, prediction, I would have equal number of each type. If they were on the same chromosome, I will have only the parental type and not the recombinant type. But my actual result gave me a lot of the parental type and a smaller unequal number of the recombinant type. This tells me they probably were present on the same chromosome and some of them crossed over, and this laid the foundation to the fact that crossing over takes place. All right. Okay, so genetic recombination is especially a product of offspring with combination of traits differing from the parents. And we know that meiosis and random fertilization or meeting of two adults or two individuals of opposite sex is totally random. So random fertilization also gives you some amount of variations. So the genetic findings of Mendel and Morgan relate to the chromosomal basis of recombination. They are basically elucidating the process of prophase 1, wherein you had crossing over and exchange of chromosomes. Okay, so this is what was actually elucidated by Thomas Hunt's experiment. Second, he also laid the foundation to genetic mapping. Okay, so the recombination frequency, how do you find out what is the frequency with which this happens? Find out how many number of recombinants you had. So they had 206 and 185. So that is 206 plus 185. And what is the total number of offsprings? All of these guys put together into 100 will give you the recombination factor frequency. In our case, it is 17%. So that means the gray body color gene and the wing shape, not gray body color gene and wing shape gene are capable of recombining at 17% recombination frequency. So that is the extent of, I wouldn't like to say distance, that is the extent to which they are linked. There can, if there is something which has got a lesser recombination frequency, that's probably in between that. Does that make sense? So for example, if I say this is 17% and now suddenly I say long proboscis and shorter proboscis is 8%, and it is like kind of in between them. So it is 8% between these guys and it is 9% between these guys, means that particular gene is kind of in between, and this is the way in which they are aligned. Okay, so you will have body color gened proboscis, which is 8%, and this is 9%, that is proboscis, and wing shape are 9% frequency. So you kind of find out where in the chromosome which gene is present. It does not tell you the distance between the two. It only talks about the percentage of frequency with which they can cross over. Okay, so this is how they found out. Now I have an explanation about how they have mapped, kind of what I explained to you 2 minutes back in the 2-3 slides back. Okay, so I just threw in this activity so we get the hang of what that particular entire topic meant. Okay, so to just run home that topic. So I have, like, say, genes A, B and C. Okay, they are located in the same chromosome, and test crosses show that recombination frequency between A and B is 28%. Now A to B is 28%. Okay, now A to C is 12%. So A to C can be 12% or it can be 12%. Got that? So there is nothing that says that it has to be on this side or on this side. And then you cannot find out the linear order of these genes till you come to know what is B to C recombination frequency. Only then you can kind of say whether it is A, C, B or an C, A, B. Yeah, that's the requirement. Only when you know the third variable you can kind of map it and kind of find the position of the genes on the chromosome as following this sequence. So another example, okay, let's walk through that. A to B is 8 and then A to C is 28. Now I do not know whether it is here, let me just call it 28, or whether it is here. I do not know right now. Okay, so then I come to know that A to D is 25. Now A to D, so I have A and D can be somewhere here and it can be 25, or A to D can be here and it can be 25. Okay, then B to C, okay, A to B was here. Okay, so B to C is 20 map units. Now B to C, if it was from here to my left side, that would be 28 plus 8, but he says it's only 20, which means the C is on this side, so it sort of confirms that this doesn't exist, or this is not what it should be. Okay, so I'm kind of saying, okay, this is my 8 and this is my B to C, which is 20. So okay, now B to D is 33. So B to D, if it is 33, it cannot be here. So this is not the place. It has to be on this side. And then it kind of falls in place wherein this 20, 25 plus this 8 gives me 33. So the sequence in which they are mapped is D, A, B, C, which means A to D is 25, A to B is 8, A to C is 28, B to C is 20, and B to D is 33. So all of these values are respected, and I am able to find out the sequence of the genes as D, A, B, C. Alright, so this is kind of the way in which they performed a series of recombinations and found out what is the outcome of the recombination frequency, with which they could find out what is the location of each of those genes on that particular chromosome. Right? This is the foundation towards gene mapping. Okay. Now we move to another topic, talking about alterations of chromosome numbers, okay, or the structure that causes some genetic disorder. Okay, so many times you have large scale chromosomal alterations that take place, which leads to spontaneous abortions or miscarriages, or cause a variety of developmental disorders. Okay, now the word non-disjunction, it's kind of like a double negative. Okay, so junction, okay, meaning joining. Okay, disjunction meaning removal of the joining. Okay, non-disjunction means not a good removal, meaning remove, it is still stuck. Where? Remember anaphase. So anaphase 1 or anaphase 2, you have the cleaving with cohesin protein or cleaving of the synaptonemal complex. And this kind of, how do I say, peels the sister chromatids apart or the homologous chromosomes apart. Alright, so if they do not separate normally during meiosis, one sperm cell or ovum will have more chromosomes than it needs, and the other one will have less chromosomes than it needs, which kind of makes both the sperm and both the sperms that are the resultant of this meiosis defective or abnormal. Right? Let's see what happens. So in this case, meiosis 1, you would have the separation during anaphase 1, wherein this non-disjunction, meaning it didn't, the homologous chromosomes did not get cleaved, or synaptonemal complex which was holding these guys here did not cleave properly. So you ended up having one cell which is, I'm sorry, just let me go, one cell which has got, instead of having 2 copies of 2 chromosomes, it is having 2 copies of 3 chromosomes, and in this case it is having 2 copies of 1 chromosome. And in this case, if it happens in the second anaphase 2, you will end up having, in this case I have 1 extra chromosome, 1 extra chromosome, 1 less chromosome, 1 less chromosome. In this case I have 1 extra, 1 less, and these may be normal because this still did a proper separation. So you see, these are the conditions wherein you can have non-disjunction taking place and leading to chromosomal abnormalities in the baby that gets fertilized, or that is the outcome of this fertilization. Aneuploidy. This word basically means fertilization of gametes in which non-disjunction or improper removal. Oh, let me just go back 1 second. So aneuploidy is improper removal. Okay, so it has not, so the chromosomes have not peeled off properly, or sister chromatids not peeled off properly. So you might have monosomic, or 1 copy of a particular gene, or you might have trisomic, 3 copies of a particular gene. So you'll see what happens in different chromosomes are higher in number or lower in number. Polyploidy is when an entire organism has 2 complete sets of chromosomes. It can be triploidy or tetraploidy. Polyploidy is very common in plants but not in animals. And interestingly, polyploids, though they have a giant kind of outcome, they are more normal in appearance than aneuploids. Okay. We also talk about, so remember, there are different ways in which you can have genetic disorders. One where an entire chromosome is more or less, which is what I showed you in the non-disjunction processes in 2-3 slides back. In this particular case, in this particular case, what has happened is a small segment, one segment or chunk of a chromosome a chromosome is extra, lost, inverted, reciprocally translocated, and you will have a ton of problems along with that. So how you would see that here is, if I label along the length of the chromosome each chunk as A, B, C, D, E, F, G, H, if there is a deletion, meaning a chromosome chunk, meaning a part is deleted, okay, and I end up losing that D gene, so that means it's a deletion. I can have a duplication suddenly, okay, I can have an inversion. So you see, it was kind of like this, now it kind of went like this. So if I had an a B and a C, it kind of twisted itself, and my C came here, A, my B came here, or my, it's like kind of the other way around, B, C, D in this case. So you see, it kind of twisted itself. Or I have reciprocal translocation. Now reciprocal translocation, you would think that this kind of chromosomal exchange takes place amongst homologous chromosomes only. So in the previous slides when I was talking about meiosis, I said M1 chromosome and D1 chromosome align themselves along the length and there is exchange. Okay, it was homologous chromosomes, but in this case, think about it, M9 and M22 kind of exchange, and that's like, or M9 and D22 kind of exchanged it, and that kind of messes up things, because now this chromosome or this chromosome, neither of them are functional in the end. Alright, they have gotten genes all mixed up. Okay, so what are the different human disorders due to chromosomal alterations? Alterations of chromosome number and structure causes lots of serious disorders. Some of the types of aneuploidy upset the genetic balance less than the other. In that, what happens is the individuals survive birthing and beyond for some time, some situations. Some of them are immediately miscarried and we don't even know why. Okay, the surviving individuals have some symptoms or a syndrome or a characteristic of a type of aneuploidy. Okay, so Down syndrome, you will have 3 copies of chromosome 21. It affects 1 out of every 700 children born in the United States. The frequency of Down syndrome increases with the age of the mother, a correlation that has not been explained. Okay, so older the mother, and you will have an increased frequency of the baby being a Down's baby, and it's kind of sort of connected towards the meiosis, which is not taking place properly. Okay, facial hair, facial features can be different. They can be short, they can have heart defects, they can have delayed development, they have an increased risk to tumors, Alzheimer's, leukemia, and sterile. But 50% of the females are sterile, 50% of the females are normal, and 50% of 100% of all the males are sterile. Okay, so in this case, if you notice, for a Down syndrome, chromosome 21, there is 3 copies of it. Okay, non-disjunction of various sex chromosomes produce a variety of aneuploid conditions. Klinefelter syndrome is the result of an extra chromosome in a male producing an individual with XXY. Okay, 1 in 1,000 males have this condition. Monosomy X or Turner syndrome produces an X0 wherein the females are sterile, but it is the only known viable monosomy in humans. It affects 1 in 2,500. Then you have trisomy X, wherein you have 3 X chromosomes. These are slightly taller and have learning disabilities. Okay, then disorders caused by structurally altered chromosomes. The syndrome called Cri-du-Chat, okay, or cry of the cat or the mule of a cat, is resulting from a specific deletion in the chromosome 5. If you look back here, the deletion, what I was talking to you, think of this as chromosome 5, and a deletion takes place, you will end up having Cri-du-Chat as your outcome. Okay, so a child born with this syndrome is mentally retarded, has a cat-like cry, and the individuals usually die in infancy or early childhood. Then you have another situation wherein a certain cancer, including chronic myelogenous leukemia, it is also called CML. It is caused between translocation between chromosomes 9 and chromosome 21. Okay, so here you have chromosome 9 and chromosome 21. Okay, there is no reason for them to have an exchange of chromosome. They do, and in the end the translocated chromosome is called the Philadelphia chromosome. The outcome is CML, or chronic myelogenous leukemia. Okay, and then a few pages on genomic imprinting. I haven't covered that for our course. A special note about Y chromosomes. It's said, remember some time back in the initial slides I was talking about Y chromosome being passed down generations unchanged. It is said that 8% of Asians are descendants of Genghis Khan, and 10% of the Irish population have the chromosome from the Irish warlord of the 5th century. And you can also trace back the Lemba tribe in southern Africa. You can trace back their origins to the ancient Jews, and they can in fact trace it to Moses' brother, it seems. Okay, interesting. So this is a chart you could try to put in. So I have a filled out one wherein you can see the answers. So you can see sex chromosomes in mammals are XX in female and XY in male. They can carry sex-linked traits. Chromosomes can also be autosomes. So if you talk about humans have 2 sex chromosomes and 44 autosomes, right? And chromosomes are present on different genes, and so they are unlinked and they have an independent assortment, and it results in different genetic recombinations. Chromosomes that are present in the genes and they are linked means they may either cross over or they do not show independent assortment. And so if they do not show independent assortment, that is like they are in the same chromosome. If they cross over, they show you a recombination frequency, which can be used to map the location of the genes. You can also have cytological evidence that to provide for to use to map and location of the genes. Chromosomes may have alterations in the number and structure, wherein you can see that some of the procedures can have errors like deletions, duplications, translocations, and inversions. And it sort of gives you an evidence, like if you look at the cell and the functioning of the cell, you can try to find out what is actually wrong and where it is wrong, and you can also map where exactly it has gone wrong. Okay, you can have alterations in the number, such as any one, which will be aneuploidy. That could be a trisomy or a monosomy, 3 copies or just 1 copy, or polyploidy, meaning an entire set. Now what I mean by that, set, meaning if you are 2n, you could just end up being 3n, or say 4n, or say 5n, or 6n. Strawberries are hexaploid, and then you will have triploid. Bananas are triploid or tetraploid. Different plants have different ploidy levels, but that does not affect them. Whereas 2n minus 1 or 2n plus 1 causes havoc in a person's genetic makeup and the outcome. Aneuploidy is worse than polyploidy. That's all for this chapter, and hope it was useful. Bye bye.